The sexual response that's characteristic of the female rat is the arching of her back.
Make sure the males are good and stimulated so that we can...
If a normal male is put with the stud, he doesn't have that response. He submits, so he doesn't have to fight. But he has no reflex arching of his back. But what Gorski has done is to find out how to give a male rat female behavior. Before puberty, there was no way the effect of that injection could be detected. But that male grew up to behave as a female. He has no male libido and behaves sexually exactly as a female would. For years, Gorski's team have been sectioning rat brains, staining them, and searching for any difference between the rats with male behavior and those with female behavior. Now they found something. This is a slice across a female rat brain, about halfway back. A feminized male would look the same. There is a tiny center that stains blue, here. In the same place, in a male brain, that center is bigger. And just recently, the mechanism by which that brain difference arises has been discovered. Precisely that sex center is dissected from the brain of a fetal mouse. Dr. Torin Alorand at Columbia University, New York, keeps that tiny fragment of brain alive in drops of liquid suspended underneath special glass slides. Half are grown in fluids to which testosterone has been added. After 8 days, this is how the brain that's growing in the presence of testosterone looks. A network of nerve fibers, rich in their interconnection. But the brain grown in the female environment is different. Only half the number of nerve fibers develop. This difference in development has only been found in the tiny sex center. Some speculate that testosterone creates the difference by preventing cell death in the male brain, but that's not certain. But the facts are that brain tissue can be made male by testosterone or female by lack of testosterone. This is true for rats. Could it be true for humans?
Now, this is a difficult question to answer. Of course, is a focus of a great deal of research. But certainly, this concept that we can change the sex of the brain is not restricted to the rat at all. It has been demonstrated in other animals, other rodents. In addition, in the primate, the rhesus monkey, and other types of monkeys. And there is some suggestion from the clinical literature that this may, in fact, apply to the human being.
In East Berlin, at the Institute for Experimental Hormone Research, is the man who is the author of that clinical literature. He believes that in the feminized male rat is the explanation of human male homosexuality. That man is Professor Gerd Dorner. His efforts to understand and cure homosexuality began 11 years ago.
One day there was a belly, a wonderful belly, and I saw the female-like behavior of the male dancers, and I had the imagination there might be a biological basis. Yes.
So in 1968, with a big research team, he began looking for a sex center, which if malformed could result in homosexuality. They began by looking for the sex center in rats. They found it before Gorski did, with this experiment. The animal is a newborn male rat. When they destroyed a small part of its brain in a very precise location, they could make the male behave as a female. In their terms, it became homosexual. What they're doing now is exploring the brains of humans. Max Schmidt is a homosexual. Professor Dorner is trying to show that humans do have a sex center, and that homosexuals have a female one. He has developed a test to show that. Max's brain will be fooled to be thinking that is in a woman's body, and Dorner will look at how it reacts to that. Estrogen will look to Max's brain like the signal from a womb after ovulation. The signals his brain sends back to this phantom womb will reveal the sex of the center in his brain. So samples of his blood are taken during the next 4 days. The amount of female ovulation-inducing hormone, called LH, is measured. Max's brain responded to the estrogen, as a woman's would, with an LH surge. That was true of 20 homosexuals. Non-homosexual males didn't react like that to their phantom wombs.
I think that is an indication that homosexual males may possess, in part, at least in part, a female differentiated brain.
He's studying mothers in their 4th month of pregnancy, the time which he believes is the critical period. Mothers' samples of fluids taken from the womb are needed for many medical reasons. To take it, the doctor first finds where the baby is, so when he samples, the needle doesn't go into the baby. Dorner gets samples from such women. He will measure the testosterone level in her womb. Should it be low, he will watch the behavior of her child if it's male to see if it becomes a homosexual. He expects to prove that low testosterone in the womb leads to homosexuality. If he does, the next step might be to inject more testosterone and so prevent it.
I think there may be the possibility in the future to prevent, at least in part, homosexuality. And that will be a question that we, for example, if we are right, that we find abnormalities in sex hormone levels, and I think we should ask the mother if we should only correct these abnormalities of sex hormone levels. And a question for the whole society, not only to me.
Dorner has made the jump from rat behavior to human homosexuality in a way which I find unacceptable.
In Dorner's experiments, he's taking a great step forward in attempting to ascertain the validity of this concept in the human. And if such experiments are done without damage or harm to the individual involved and under controlled conditions, then the work that Dorner is doing could give us a solid answer whether or not there is a dependence on hormones of homosexual behavior. I think the verdict is not in yet.
But while in Berlin, Professor Dorner showed us some of his research that is so far unknown to Western scientists. For 2 separate hours each day, pregnant rats are subject to stress. The stress is being unable to move while exposed to strong arc lamps. Animals under stress produce adrenaline. Adrenaline suppresses the action of testicles. So perhaps the male fetuses in these rats have lower testosterone levels than normal.
Now we have done many experiments during the last weeks and months, and we could clearly show that there is a clear decrease of the testosterone in the fetuses and the newborns after stress of the mother animal. And there is also the possibility in the humans, and we have looked for the frequencies of male homosexualities which were born during the especially stressful situation in Germany during war, and have compared them with those which were born before, 6 years before and 6 years after war. And in about 500 cases, we could find that about twice as many male homosexuals were born, indeed, during war as compared to the years before or after war. That may be an indication that stress may be important for permanent changes of sex hormones or other hormones, and may induce permanent changes of the fetuses and of the offspring.
The evidence from the hermaphrodite in Cape Town does not support Dorner's theory. Mr. Blackwell has a female brain, so female that he used to ovulate once a month.
Do you have a girlfriend?
Yes.
And this makes you happy? Do you look forward to seeing her again?
Yes.
Good. It's really fascinating if you consider the fact that his whole hormonal profile is completely female, yet he has a normal psychosexual behavior of a boy. And that, I think, is fascinating, because it shows that during fetal life, his hypothalamus didn't androgenize as far as the hormonal production is concerned. And yet he has this normal male psychosexual behavior. And this, I think, is a very